basically be a review. This is basically the innervation and the neuroregulation of smooth muscle. So this is happening via the autonomic nervous system. The divisions here are sympathetic and parasympathetic. Now this can be excitatory or inhibitory. Now remember, we talked about the fact that skeletal muscle is always excitatory. Here, it can be excitatory or inhibitory. And just to kind of draw an example from what we talked about before, we said that all alpha receptors bring about an excitatory event to their tissues, whereas beta-2 receptors, for example, bring about an inhibitory effect to their tissues. So the response at the tissue level is not always excitatory the way that it is with skeletal muscle. The precise response will depend on what type of receptor, again, whether it's alpha, beta, the different types of beta, alpha 1, alpha 2, what type of neurotransmitter is released is also going to play a crucial role as well, whether it's that acetylcholine at the parasympathetic postganglionic junction, or it is norepinephrine at the sympathetic postganglionic or neuroeffector junction. We've also got the presence of varicosities here. So this is something we know we've talked about before. At the neuroeffector junction, we always see those swellings along the nerve ending, which are varicosities, which release the neurotransmitter that's responsible for this type of signaling or innervation to smooth muscle. And then the diffusion of that neurotransmitter and the binding to these receptors is what actually brings about the muscle contraction that we see on smooth muscle. All right, let me pause here just a bit and see what questions you've got. So we talked about the structure of smooth muscle, and I keep interjecting with, differences between smooth and skeletal or between smooth and cardiac. It's really important for you to be able to discern some of these properties and features that are shared or that are, dissimilar between these types of muscle. All right. So let's keep going here. So we'll talk now about the classification of smooth muscle. So there are actually two different types of smooth muscle, and they kind of behave differently. And so when you draw your Venn diagram, I'm going to encourage you to draw, a large circle for cardiac muscle, a large circle for skeletal muscle, and then a large circle for single unit smooth muscle, and another large circle or sector for multi-unit smooth muscle. Because they're kind of similar in that they're both smooth muscle, but then they do have some very obvious and important differences. So single unit smooth muscle behaves just like what it sounds like, one entire single unit of muscle. Whereas multi-unit smooth muscle acts like different muscle cells that are behaving and that are regulated individually. And so we're going to go on to talk about some of the differences of those types of muscle here. So single unit smooth muscle is the most common, the most abundant type of smooth muscle. It's found in the GI tract. We also see it in blood vessels, particularly smaller blood vessels. And then we also see it in the respiratory tract as well. The tissues here function synchronously, they function together. And I want you to kind of think about all these little smooth muscle cells pretty much holding hands, and the hands, so to speak, that they're holding are these gap junctions. So the gap junctions are basically connecting these smooth muscle cells together. And this is what's responsible for this type of muscle acting like one synchronous unit. And so when we have contraction of one single unit cell, the entire muscle will contract together. There's no, recruitment, there's no individual or, customized contraction that's seen with a single unit type of smooth muscle. So we've got gap junctions found throughout this type of muscle. There's pacemaker ability, that is the ability to spontaneously depolarize. So I want to explain what this pacemaker potential, this pacemaker ability is. It's only seen in single unit smooth muscle and cardiac muscle. It's basically the ability of this type of muscle to spontaneously depolarize. So whereas we saw in skeletal muscle that it had to be innervated, there had to be that neurotransmitter crossing the junction, binding to a nicotinic receptor, bringing about an end plate potential, here we can see the contraction of this muscle in the absence of any type of neural stimulation, in the absence of any type of neurotransmitter or chemical stimulation. And that's what we call pacemaker ability or pacemaker potential. The innervation is to a few cells, so not each cell is receiving innervation. And if you look at this schematic up here, we see our postganglionic neuron. So remember that this is that second neuron in series, right? Making that connection back to the autonomic nervous system. It's got the varicosities, right? So this is essentially the neuroeffector junction. And so the varicosities that have the swellings with the neurotransmitter is going to diffuse across, but it's not going to diffuse to every single cell. It's only going to diffuse to a couple of cells. And because they're kind of holding hands, so to speak, right, because of these gap junctions, the depolarization, the excitation of a few cells will eventually trickle down or travel to all of the cells, and that will allow this entire muscle to contract as one synchronous unit. Because of the pacemaker potential, pacemaker ability of single unit cells, they have what's called tone. And tone is this level of contraction that's happening without any type of stimulation. So on its own, and we see tone particularly in blood vessels, so that's a really good example of where we see tone. When blood vessels are not being innervated or stimulated, they're still slightly contracted. Right? They're not just completely relaxed the way that some other muscles are. They're still slightly contracted, irregardless of not being stimulated from any type of external neurotransmitter or neuron. We can increase and decrease the tension here because of the fact that we have a single unit. We don't see any recruitment. We've got this stretch reflex that we see with single unit smooth muscle. So that means that sudden or prolonged stretch actually has this relaxation response. And then again, we see graded contractions here, meaning that there is no recruitment because everything is acting as one unit. There is not this ability to call on additional cells. Everyone is already kind of at the party, if you will. And we can't call on additional fibers to offer more force. We see varying levels of intracellular calcium that will dictate the increases and decreases in tension. So whereas in skeletal muscle here, and I apologize, I keep kind of going back to skeletal muscle, I hope I'm not confusing you all, but whereas in skeletal muscle, we saw that in order to increase the amount of tension or force that skeletal muscle produces, what we did was call on or recruit additional fibers to produce more force, right, or more cross bridges. In smooth muscle, because specifically single unit smooth muscle, because all of the cells are acting as a single unit, we cannot recruit additional cells, right? Everyone is already contracting as one unit. So the way that we increase and decrease tension is by manipulating how much intracellular calcium is being released into, or basically manipulating how much calcium is either coming in from the extracellular environment or being released from the sarcoplasmic reticulum. And so the varying intracellular levels of calcium are what dictate the amount of force or tension that is produced by this muscle and not recruitment. I hope that made sense. All right, so that was single unit smooth muscle, very similar to cardiac muscle in that it's got gap junctions, also similar in other ways to skeletal muscle, right? Let's talk about multi-unit smooth muscle. So some of the areas that we see multi-unit smooth muscle are the large airways, arteries, particularly larger elastic arteries, muscles of the eye, such as the ciliary muscle, muscles of the iris, and then some of the larger parts, larger parts of the airway. Now, there are very few gap junctions here, so not a lot of gap junctions. And that is to facilitate each of these smooth muscle cells acting as their own individual unit, if you will. So each fiber gets innervated individually. And so we can see a different setup here. We see these varicosities once again, and these postganglionic neurons, but they're kind of more interweaved within the muscle cells themselves. And so each muscle cell is going to be receiving its own diffusion of neurotransmitter from the vesicles. And so we don't have the gap junctions that are kind of facilitating the synchronous operation the way that we saw with single unit smooth muscle. So each fiber receives its own innervation. We don't see tone here because these cells do not have pacemaker potential. That is, they cannot spontaneously depolarize on their own. And then again, we can see recruitment here because each individual cell is acting as its own unit. So if I've got 3 cells that are contracting and I want to produce more tension or more force, because each of the cells are acting as their own boss, their own unit, so to speak, I can recruit another cell, I can recruit another cell, and so on, and I can increase the amount of tension or force that I'm producing that way. Right. And here's a kind of summary diagram, which is pretty much everything that I just said, but just the way to, draw it out on your own and visualize it on your own. So I encourage you to draw this out, and I'll make these drawings available on Brightspace in the event that you want to compare them to what you drew on your own. All right, so the final thing we're going to look at here is this idea of pacemaker ability or pacemaker potential. And so basically we've got this phenomenon that's seen again with single unit smooth muscle as well as cardiac muscle. We're going to talk about this pacemaker ability in cardiac muscle as well. But we've got these slow wave potentials. So there are these spontaneous cyclical changes in the membrane potential. And so these slow wave potentials are basically not strong enough to actually incite an action potential the way that a pacemaker potential is, but these types of cyclical changes in the resting membrane potential are what get us closer to actually bringing about a pacemaker potential. So here for these, slow wave potentials, we basically have this cyclical, spontaneous change to the membrane potential in terms of sodium. So we got cyclical, changes to the membrane permeability for sodium. And that kind of makes sense, right? We know that sodium is one of the main ions that can rush into the cell and bring about a positive membrane potential change or help to depolarize the membrane potential. And so when we have this cyclical change in the membrane's permeability to sodium, we kind of have these brief periods of sodium rushing into the cell, not enough to get us to threshold, but just enough to give us a cyclical wave in the resting membrane potential. And that is a slow wave potential. Now, slow wave potentials can contribute to a pacemaker potential. And a pacemaker potential is an actual spontaneous depolarization where we get to threshold. And I'll talk about how threshold value kind of differs with smooth muscle. It's around 50 for smooth muscle, and it's around 70 for cardiac muscle. So it's kind of different than what we saw for some of our other muscle and for neurons. But it's enough of a change to get us to our threshold, and then we can fire a spontaneous action potential. So this is happening without any neural stimulation, without any neurotransmitter or chemical stimulation. This is happening completely spontaneously. And so the types of membrane permeability changes that we see that result in these pacemaker potentials is we can have an increase in the permeability for calcium. We know calcium is also a positively charged ion, and if we increase its permeability, it can rush into the cell and help to bring about a spontaneous depolarization. We can also see an increase in the permeability to sodium. We'll do the same thing, right? 2 positively charged ions. If they rush into the cell, this can spontaneously get us to fire an action potential, or we can see a spontaneous decrease in the membrane's permeability to potassium. And we know that potassium is also positively charged, but it has the opposite ECDF. So it's going to try to get out of the cell. And so if we let potassium leave the cell in excess quantities, that ends up being a depolarizing event, which will also be more likely to get us to threshold and to spontaneously depolarize the cell. Okay, so these types of events can contribute to the spontaneous depolarization that we see with single unit smooth muscle, and eventually we'll talk about it with cardiac muscle as well. Okay. So the last thing we'll talk about here is cardiac muscle. This is a perfect segue to getting into the cardiovascular system that we're gonna start the next time we meet. So the cardiac muscle features are kind of similar to skeletal muscle. They're striated, so they've got those sarcomeres. We see the troponin and tropomyosin regulation that we also saw with skeletal muscle, so not the calmodulin regulation that we see in smooth muscle. We see gap junctions here. So in cardiac muscle and in a single unit smooth muscle, we see gap junctions. In cardiac muscle only, we see these intercalated discs. And I'll talk about what these intercalated discs are when we get to cardiac muscle, but they're basically these junctions between cardiac muscle cells where these gap junctions are present. We also see this pacemaker ability, the ability to spontaneously depolarize these cells, the way that we just saw with that single unit type of smooth muscle. Here, the innervation is also involuntary, so it's going to be the autonomic nervous system innervating this type of muscle, but for obvious reasons. We see calcium coming from the extracellular fluid and the SR, the way that we saw with smooth muscle. And then we see action potentials lasting almost as long as tension does. And so because the action potentials are lasting as long as the tension is for cardiac muscle, there's not an ability to sum. We cannot have trap. We cannot have summation, the way that we saw with skeletal muscle, because the action potential is about the same length in time as the actual muscle contraction is. And so we've got that refractory period here. And so there's a time within that period of a single cardiac muscle contraction where we cannot physically fire another action potential because of this refractory period that's almost the exact same length of a muscle contraction to begin with. And so because of that phenomenon and that makeup, we cannot have summation, we cannot have trap, we cannot have tetanus with cardiac muscle. And I'm sure you're imagining why that is a protective mechanism. All right. Now the final slide here has a very nice summary table. It's got features of skeletal muscle, features of single unit, multi-unit, and cardiac muscle. Again, I encourage you to either draw this table out or draw a Venn diagram out where you can actually point out some of the shared features. But point out especially the regulatory aspects of these types of muscle. The fact that the regulatory protein in skeletal muscle and cardiac muscle is troponin, but in smooth muscle, it's calmodulin. The sources of calcium, right? For cardiac and smooth muscle, it's also bringing calcium from the extracellular fluid environment. Which of these muscles have gap junctions? Which of these muscles have pacemaker potential? Which of these muscles can exhibit recruitment, right? Only skeletal muscle and single unit, multi-unit smooth muscle can exhibit recruitment. So it's really important for you to have all of these differences, these different features down in which distinguish between these different types of muscle.